Oncostatin M. Oncostatin M, also known as OSM, is a protein that in humans is encoded by the OSM gene. OSM is a pleiotropic cytokine that belongs to the interleukin 6 group of cytokines. Of these cytokines, it most closely resembles leukemia, inhibitory factor LIF in both structure and function. As yet poorly defined, it is proving important in liver development, haematopoeisis, inflammation, and possibly CNS development. It is also associated with bone formation and destruction. OSM signals through cell surface receptors that contain the protein gene.130. The type I receptor is composed of gene.130. Discovery, isolation, and cloning. The human form of OSM was originally isolated in 1986 from the growth media of PMA-treated human S937 histiocytic lymphoma cells by its ability to inhibit the growth of SAE. A robust protein, OSM is stable between pH 2 and 11 and resistant to heating for 1 hour at 56 deg C. A partial amino acid sequence allowed the isolation of human OSM cDNA and subsequently genomic clones. The full cDNA clone of HOSM encodes a 252 amino acid precursor, the first 25 amino acids of which functions as a secretory signal peptide, which on removal yields the soluble 227 amino acid pro-OSM. Cleavage of the C-terminal most 31 residues at a trypsin-like cleavage site yields the fully active 196 residue form. 2 potential and glycosylation sites are present in HOSM, both of which are retained in the mature form. The 196 residue OSM is the predominant form, isolated form, a variety of cell lines, and corresponds to a glycoprotein of 28 kDa, although the larger 227 residue pro-OSM can be isolated from overtransfected cells. Pro-OSM, although an order of magnitude less efficacious in growth inhibition assays, displays similar binding affinity toward cells in radioligand binding assays. Thus, post-translational processing may play a significant role in the in vivo function of OSM. Like many cytokines, OSM is produced from cells by de novo synthesis followed by release through the classical secretion pathway. However, OSM can be released from preformed stores within polymorphonuclear leukocytes on degranulation. It still remains unclear how OSM is targeted to these intracellular compartments. Structure. Primary sequence analysis of OSM allocates it to the gene.130 group of cytokines. OSM most resembles LIF, bearing 22% sequence identity and 30% similarity. Incidentally, the genes for OSM and LIF occur in tandem on human chromosome 22. Both LIF and OSM genes have very similar gene structures, sharing similar promoter elements and intron-exon structure. These data suggest that OSM and LIF arose relatively recently in evolutionary terms by gene duplication. Of the 5 cysteine residues within the human OSM sequence, 4 participate in disulfide bridges. One of these disulfide bonds, namely between helices of B, is necessary for OSM activity. The free cysteine residue does not appear to mediate dimerization of OSM. The 3-dimensional structure of human OSM has been solved to atomic resolution, confirming the predicted long-chain 4-helix bundle topology. Comparing this structure with the known structures of other known LC cytokines shows it to be most closely related to LIF RMST of 2.1 across 145 equivalent Ca. A distinctive kink in the helix arises from departure of the classical alpha helical H bonding pattern, a feature shared with all known structures of LIFR using cytokines. This kink results in a different special positioning of one extreme of the bundle to the other, significantly affecting the relative positioning of site Roman 3 with sites I and Roman 2C, receptor recruitment sites. Receptors. Receptor recruitment sites. Oncostatin M triggers the formation of receptor complexes by binding to receptors via 2 binding sites named site Roman 2 and site Roman 3. The nomenclature of these sites is taken by direct analogy to growth hormone, probably the best studied of 4-helix bundle cytokines. Site Roman II consists of exposed residues within the NC helices and confers binding to GP130. The crucial residues of site Roman III are located at the N-terminal extremity of the D helix. This site is the most conserved amongst IL-6-like cytokines. OSM contains a conserved phenylalanine and lysine residues Pho160 and Co163. Cytokines that recruit LIFR via site 3, i.e. LIF, OSM CNTF, and CT-1 possess these conserved phenylalanine and lysine residues and is known as the FK motif. Signal transduction through OSM receptors. Signaling by type I and type Roman II OSM receptors have now been shown to be qualitatively distinct. These differences in signaling character, in addition to the tissue distribution profiles of OSMR and LIFR, offer another variable in the distinction between the common and specific cellular effects of OSM with respect to LIFR. All IL-6 cytokines, whether they homo or heterodimerize GP130, seem to activate YA-CoN, YA-K2, and to a lesser degree TYK2. YA-CoN, YA-K2, and TYK2 are not interchangeable in the GP130 system. This has been demonstrated with the use of YA-CoN, JAK2 or TYK2 deficient cell lines obtained from mutant mice. Cells from YA-CoN deficient mice show reduced STAT activation and generation of biological responses in response to IL-6 and LIFR. In contrast, fibroblasts derived from YA-K2 null mice can respond to IL-6 with demonstrable tyrosine phosphorylation of GP130, YA-CoN, and TYK2. Thus, it seems YA-CoN is the critical JAK required for GP130 signaling. Activation of the same JAKs by all three receptor combinations GP130/GP130, GP130/LIFR, GP130/OSMR raises the question of how IL-6, LIF, and OSM can activate distinct intracellular signaling pathways. Selection of particular substrates, i.e. STAT isoform, depended not on which JAK is activated, but instead are determined by specific motifs, especially tyrosine-based motifs, within each receptor intracellular domain. Aligning the intracellular domains of GP130, LIFR, and OSMR results in some interesting observations. Sequence identity is generally quite low across the group, averaging at 4.6%. However, as with many class I haematopoietin receptors, two short membrane proximal motifs, termed box 1 and box 2, are present. In addition, these receptors also contain a serine-rich region and a third more poorly conserved motif termed box 3. Box 1 is present in all signaling cytokine receptors. It is characteristically rich in proline residues and is essential for the association and activation of JAK. Box 2 is also important for association with JAK. GP130 contains box 1 and box 2 sequences within the membrane proximal part of the cytoplasmic region, lying within the minimum 61 amino acids required for receptor activation. Mutations within the boxone region reduce the ability of GP130 to associate with JAKs and abolish ligand-induced activation of JAK1 and JAK2. Box 2 also contributes to activation and binding of JAK. Studies with various GP130 truncation mutants show a reduction of JAK2 binding and abrogation of certain biological effects upon deletion of box 2. However, JAKs are able to associate with GP130 devoid of box 2 when overexpressed. LIFR and OSMR also contain the membrane proximal boxone/box 2-like regions. The first 65 amino acid residues in the cytoplasmic domain of LIFR, in combination with full-length GP130, can generate signaling on treatment with LIF. Co-precipitation of JAK1, JAK2, and TYK2 with receptors containing cytoplasmic parts of the LIFR or OSMR. All beta receptor subunits of the GP130 system also possess a box 3 region. This region corresponds to the C-terminal amino acids of the OSMR and LIFR receptors respectively. Box 3 is necessary for the action of OSMR However, BOX3 is dispensable for the action of LIFR. In the case of GPO130, BOX3 is dispensable for activity. However, the presence of an intact BOX3 sequence is required for certain aspects of GPO130 signaling, i.e. stimulation of transcription, through the STAT-3 response element. In addition to the poor sequence conservation amongst the intracellular domains of GPO130 receptors, the number and position of conserved tyrosine residues are also poorly conserved. For example, LIFR and OSMR share 3 homologous tyrosines. In contrast, none of the tyrosine residues present in the intracellular domain of GPO130 share equivalents with LIFR or OSMR, even though the intracellular regions of LIFR and GPO130 share more sequence identity than LIFR and OSMR. Of the proteins recruited to type I cytokine receptors, STAT proteins remain the best studied. Haematopoeisis of GPO130 has been shown to phosphorylate and activate both STA-tone and STAT3. GPO130 preferentially activates STAT3, which it can do through 4 STAT3 activation consensus sequences IXC, IFC, A905 A915 imp. The lower propensity for STA-tone activation may be a reflection of the lower number of STA-tone activation sequences, where X is any residue and Z is any uncharged residue, namely A905 and A915. Cytokines that signal via haemotomeric complexes of LIFR or OSMR, i.e. devoid of GPO130, are currently unknown in nature. However, various investigators have attempted artificial haematomerization of LIFR and OSMR intracellular domains, with conflicting results, by constructing receptor chimeras that fuse the extracellular domain of one cytokine receptor with the intracellular domain of LIFR or OSMR. Signaling by LIFR intracellular domain haematomerization has been demonstrated in hepatoma and neuroblastoma cells, embryonic stem cells, and COS-1 cells by using chimeric receptors that haematomerize upon stimulation with their cognate cytokines, i.e. GCSF, neurotrophin-3X. However, a GCSFR/LIFR chimera was not capable of signaling in MON or BAF cells. Anti- or pro-inflammatory. The role of OSM as an inflammatory mediator was clear as early as 1986. Its precise effect on the immune system, as with any cytokine, is far from clear. However, 2 schools of thought are emerging. The first proposes that OSM is pro-inflammatory, whilst the other holds the opposite view, claiming OSM is anti-inflammatory. It is important to note that before 1997, differences in human and murine OSM receptor usage were unknown. As a result, several investigators used human OSM in mouse assays, and thus any conclusion drawn from the results of these experiments will be representative of LIF, i.e. signaling through GPO130/LIFR complexes. OSM is synthesized by stimulated T cells and monocytes. The effects of OSM on endothelial cells suggest a pro-inflammatory role for OSM. Endothelial cells possess a large number of OSM receptors. Stimulation of a primary endothelial culture HUVEC with HOSM results in delayed but prolonged upregulation of P-selectin, which facilitates leukocyte adhesion and rolling necessary for their extravasation. OSM also promotes the production of IL-6 from these cells. As mentioned above, the action of OSM as a quencher of the inflammatory response is by no means established yet. For example, conflicting results exist as to the action of OSM on various models of arthritis. For example, OSM reduces the degree of joint destruction in an antibody-induced model of rheumatoid arthritis. OSM is a major growth factor for Kaposi's sarcoma spindle cells, which are of endothelial origin. These cells do not express LIFR but do express OSMR at high levels. For example, OSM can modulate the expression of IL-6, an important regulator of the host defense system. OSM can regulate the expression of acute phase proteins. OSM regulates the expression of various protease and protease inhibitors, for example gelatinase and AN chymotrypsin inhibitor.